No niin, morjes Mesimestarilta taas. On täällä pohdittu mehiläishoitajia hirveästi pohdituttaa ja meitäkin, että mikä juttu se on se uusi DNA-testaus Hunajalla ja sieltä huhutaan, että olisi paljon väärennöksiä kauppojen hyllyillä ja se on uusi menetelmä ja kukaan ei tunnu tietävän sitä kauheasti mitään, niin mehän päätettiin Konstan kanssa ottaa asiasta selvää ja lähteä käymään pikaseen Tartossa. Meillä on siellä tuttuja mehiläishoitajia ja asiakkaita ja laboratorio sattuu sijaitseen siellä ja tostahan kulkee lauttoja, että muutamalla satasella kaikki matkakustannukset jo peittyi ja oikein yllätyttiin positiivisesti, koska meille esiteltiin 2,5 tuntia sitä laboratorioa ja sen toimintaa. Tehtiin siitä 0,5 tunnin tiivistelmävideo, eli luento, miten se homma toimii ja mitä kaikkea siinä näkee. Ja näin ollen, jos oikein kiinnostaa, niin varaa 0,5 tuntia ja katso tämä video, niin tiedät paljon enemmän siitä Hunajan DNA-testauksesta. Mitä te näette siitä DNA-testistä? Mitä se tuo näkyviin oikeasti? Miten siitä tämä kaikki tulee esille?
If we study the honey total DNA, then based on that analysis, we see the full spectrum of species. It means like majority of the species are plants. Then one very important sector is microbes and microbiota. Then what else? Yeast, because yeast are important part in the hive, because yeast are everywhere. Then what else? We see the insect, the honeybee itself. Then we see the DNA viruses, because that is the part of honeybee's life. Even we see your DNA, your cat DNA, your dog DNA, and cow DNA. It means the mammalians. And others as well. So this picture, it is like a highly complex, but it is very unique to authentic honey. All the sectors should be in balance, and that is like a normal picture of the organism. The honey hive is like a big organism or like a small ecosystem. So those should be in the balance between the plants, microbes, yeast, and insects. And even we should see the viruses and the mammalian DNA. So that is the normal picture of authentic honey, and that's what we see.
So what can we see about your report and what this number means?
Yes, the number means that if you study your sample, if the score is 100, for example, it means that it is very, very similar to the authentic honey database. Yeah. And we can draw the authentic honey database as a cloud. It's somewhere here. And it contains many honeys in many different countries. And they are all authentic ones. And if we test like an unknown sample, then the unknown sample can be here, here. I make another color. The unknown sample can be here, 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 or here, based on the computational analysis. Mm-hmm. Now, what the analysis says, if the score is 100, it is in the middle of this authentic honey group. It means it is the member of authentic honey. And then the score is 100. Uh-huh. There is of course deviation, the normal deviation in the group. The group is not the one dot only. Uh-huh. If the normal deviation is here, then the score is 55, for example. It is still member of this group. Okay. Yes. If the score is 10, that is the scale from 0, 10, 55 and 100. As higher is the score, it means the central point of the group. If the score is 80, for example, it doesn't mean that the 20% is syrup.
Okay.
It just means that based on the analysis, it is a bit distant than the central point. Yes, 55 can be also authentic honey, but there is some normal deviation. This is definitely fake honey, and this is definitely fake honey, and that is the score.
But can we say that 54 is fake honey, 55 is honey? No.
Unfortunately, no. If there is a borderline case, for example, 50, then we consider all parameters. We see the data like carefully, human take a look, because of course there is some plus and minus.
Okay.
Yes, and then we see the markers of the fake honey. We know the markers of fake honey, and then we make the final decision what is the status of this 50, for example.
Yeah. And if something honey is real fake, here is number one, but have sugar syrup what Chinese put inside honey, is inside this sugar syrup something special DNA what you can see?
Yes.
Yes.
Yes, the sugar syrups are not clean. They are produced from some raw material. It has the clear traces of DNA. If it comes from the rice, if it comes from corn, for example, and so on. Other components are also added into this fake honey. It is not pure syrup. Because if you add only pure syrup, you do not get such consistency. You need something what make it more viscous first. Then you need the component of the color, because syrup is transparent. And if such components are involved, then we detect it based on the DNA analysis. And of course the balance, the same. The balance between the microbiota, yeast, plants, viruses, and so on. So the full picture should be according to the authentic honey.
Okay.
Yeah.
That's fine.
So what was the case? Like there is 48. What was the case? And it wasn't real.
We checked the markers of fake honey, and we saw the proportion increased there. It is just less diluted, let's say so.
Yeah.
And that's why we decided that 48 it is still fake, because we saw the clear markers of fake honey.
Oh, okay.
For example, if that is 48 and that is 8, it means that the increase of fake honey markers were here much higher than here. So it was like a bit less diluted, maybe. We don't know what happened. We do not make police work here. We just see the outcome of the tested jars.
So was that 48 like better fake?
Probably, or less diluted.
Yeah.
Or less diluted, but definitely diluted, or definitely non-authentic honey.
Yeah.
That is for sure.
Yeah.
But have you honey samples from other
Yes.
places too?
Yes.
South America and Africa and Australia.
Yes, they are common, but the main of this was that was the EU project,
Yeah.
and the EU supported project.
Okay.
But definitely we should focus first for EU, because if you, for example, ask authentic honey samples from China, there is no chance to get any authentic honey samples, because their culture is produce the mass. And it is okay for them to feed the honeybees with the syrup, different kind of syrup, and even then to add like pigments or whatever sugars or syrups what are needed for commercial purposes. Of course, if somebody produces for them self-purpose, then no point to cheat. But for commercial purposes, yes, they follow the procedure to produce more and more and more. So there is no chance to get the authentic ones. We have some, but I'm still skeptic.
Okei.
Yeah.
Okei.
The lab part starts here. Lab assistants are here in the open office.
Okay.
That is the start of the lab. And let's go to the research front.
Tere.
That is the start of the blood samples, because we accept the blood samples of the pregnant woman.
Okay.
If the woman is pregnant, then they give the blood sample, and our duty is to screen out the chromosomal diseases, like Down syndrome, for example, and the other like nasty diseases what may happen.
Okay.
And that is from the blood sample. Actually, we do the same. We do the total DNA sequencing. Like whatever is in the maternal blood sample, then we sequence it and see that yes, in the fetus there can be like mistakes, the chromosomal diseases, and then we see it. It is not only for Down syndrome, for example, but there are like all possible chromosomal diseases over the genome. So we use the same technology in honey.
Wonderful.
Everything what is inside the honey, we do the same procedure basically. But yes, the morning samples haven't arrived yet. It is too early, but otherwise the samples come on the table, and then they start to unpack and start the process. There is one instrument for the DNA extraction, that is the robotics,
Okay.
and yeah, general things.
Sen verran korkeita tekniikkaa, että menee osa yli hilseen muunkin kuin kielitaidon takia.
Siellä ne pystyy aikamoisia.
So this instrument is called as a DNA sequencer.
Okay.
It is 130 kilo of computer and lasers and scanners, basically. And yes, it is able to, this one instrument is able to analyze, for example, all Estonian pregnant women. So the capacity is pretty high for this tiny instrument. We analyze here pregnant women samples, other medical samples and honey samples.
Okay.
It eats everything because we prepare the samples in different way, but finally they will be sequenced here, and that gives us DNA data out.
Hunaja on päässyt nyt kyllä vähän korkeampaan seuraan kuin normaalisti.
Yep. And that is also general lab, nothing special, just clean space for every project. Now let's go. There is basically nothing. Let's go to the honey lab. So that is short-term storage. We store the commercial ones and the latest samples. For example,
Mitä löytyy?
yeah.
Osa suomalaisista. Not
Yeah.
all.
Not all.
Tuossa on lisää.
Other boxes.
Yeah.
Here is 2 boxes more.
Yeah.
Honey from
boxes.
Finnish.
There is more.
You
Yeah.
mean that one?
Yeah.
Yeah, why this one Finland too? Yeah.
And we basically label the same. The label means that is past label. It's sticked here. You see?
Uudenmaan hunaja. Voidaanko näyttää? Can we show?
Yeah.
Uudenmaan hunaja. Siinä on numero 100. Eli tämä vastaa täysin hunajan kuvaa niiden DNA-kartassa.
Yeah.
Se on aitoa.
Let's take another example. Also Finnish honey.
Joo.
Nice score.
Suomalainen luonnontuote mesikämmen hunaja 93%. Ei ole prosenttia, anteeksi, vaan 93
Yeah.
numero, eli se on, sijaitsee siellä DNA-kartalla kohdassa, jos 100 on keskellä, niin tämä on sitä hiukan syrjää, mutta tämä on niin kuin täysin autenttista hunajaa.
And what's here? Haha. Maribel. Little
Maribel.
Maribel. The sticker color means it is atypical,
Yeah.
and the score is 16, which is under the threshold.
Not even close.
Not even close. Not even close. And another Finnish sample.
Voi hyvin hunaja.
Yeah. Also example. Also
84.
Nice. Yeah.
Eli se ei ole niin kuin siinä keskiarvossa 100, mutta se on sen sisällä, sen normaali vaihtelun sisällä ja on normaalia hunajaa.
Exactly.
Yeah.
So that is the example. So and that is the lab. That is the lab. Not big, not small. Right now it's enough. Let's take 2 samples. No point to show the labels, but generally it is the life. We put honey to that level, 20 ml, and that is basically enough. And another sample. Now we make the syrup. We add water. We dissolve it a bit, and we start then the extraction of DNA. Now we have to dissolve. Adjust it for honey procedures. Just to be sure. No, still. We capture all like solid pieces from this syrup, and they are forming the dot in the bottom. You will see in 10 minutes.
Yes.
And that is the source of DNA extraction. Now we have 10 minutes. But after that, you will see.
Yeah.
So let's take the samples out. And you see, this is the material for DNA extraction. Like this tiny dot here and here.
How many DNA is in this
Enough.
sample?
Always enough. So yeah, we need actually so little DNA. And in normal honey there is always DNA. In case of other manipulated liquids, it may happen that there is no DNA. Yeah. But soon I let them dry a bit, and then we start the next step. So basically, if that is probably non-authentic honey, it doesn't mean that this contains more DNA or it is like a more authentic because it has more material. No. It may mean that there are some additives that change the color and the content of the honey.
Hey.
So it is. It may not be like a natural product of the honeybee. So, and we start the DNA extraction procedure. We dissolve it. And put it here. So that is one of the manual work, because the scaling is manual. If we are working with the big tubes, then it's manual, but later on with the small tubes, it's easier to automate and make the life easier in the lab.
Okay.
So it's not so tricky anymore. But yes, generally they are ready for DNA extraction in the later. It's time to show the results, and Kairi can explain you the results. Like example of what we see, the full picture, and you can use the same in later on in the video. You explain the plants, microbes, yeast, and so on.
This is only one example of or one authentic honey, how the DNA profile looks like.
Yeah.
And I don't know what you were talking about, but usually the honey contains DNA from various groups of different kind of organisms.
Yeah.
And this is like the DNA profile that describes what kind of plants or bacteria or other organisms we can see in the DNA profile. Usually most of the biggest part of honey DNA comes from different kind of plants.
Mm-hmm.
Here is one example. It can vary. There can be like different kind of samples, but this is one example. From the hive there was also American fault brood infection.
Mm-hmm.
And we can see different kind of plants. This is not the whole picture. Actually, we can see thousands or hundreds different kind of plant species. This you can see more, 18 more or 5 more. You can like zoom in different kind of orders, plant orders, families, genus. Okay, we can zoom in and actually we can see different kind of plants more. This is Latin.
Here come.
Yeah.
Hey.
Yeah.
This small scale here is.
Yeah. You can like see zoom in and you can click here and see more with different kind of orders, different kind of plants. So you can. This is the whole picture, the like raw data that we can see. What kind of plants? How many DNA sequences goes to what kind of plant species?
Total.
This is
1,860,000.
Yeah, goes to the plants.
Oh.
Actually, as a result of our analysis, we can get like 10 to 20 million DNA sequences for different kind of organisms, for plants, for bacteria. So this is only plant part. Okay, the whole here is the whole results of DNA sequences. It's 4 million, more than 4 million reads or DNA sequences in this sample. And yeah, about 70% goes to the plants. Then we can see different kind of bacteria. Most of the bacteria comes from the microbiota of honeybee. Also bacteria from the environment, from honey production environment, from flower environment, plant bacteria related or plant disease related bacteria. Also bacteria that causes some kind of infections in honeybees, like American fault brood causing bacteria we can see. And different other, we can like also zoom in what kind of bacteria. The most abundant is like Apilactobacillus concuncate, that shows the health of honeybees. It's a good bacteria.
Okay.
From microbiota. And this is bad bacteria causing American fault brood.
Oh.
Yeah, and different other from environment related to the plants or related to the honeybee. So the picture is actually wider that we can see about bacteria. but the picture changes. If you do something, if you are feeding your honeybees with syrup or doing something like that, it changes the picture.
Okay.
And if you add some syrup, which also contains DNA from environment, it changes the picture. And yeah, this is like fingerprint. It describes very well the environment where the honeybees have been, the flower environment where honeybee has flown through, the hive environment, and then also the honey production environment. So it's very well describes the whole process. And then the authentic honeys, there are like similarities between, if you're looking different kind of authentic honeys, there are similarities between authentic honeys, and there are some differences between authentic and non-authentic honeys, and specific there can be like different differences between different kind of elaborations. And so we are using like machine learning and technology to distinguish the authentic honeys and non-authentic honeys, because we are using the whole picture like this, not targeting any specific orange scopes or something, but analyzing the whole picture. Is it authentic or not?
Tämä kaikki tieto tulee siitä alle nuppineulan pään kokoisesta nökäleestä, mikä jäi siitä hunajanäytteestä sinne muoviaskin kärkeen. Ihan ihmeellistä. Me ollaan sokeita, me ihmiset, me ei nähdä mitään noin pientä asiaa.
Like in criminalistics and medicines, they are using like DNA-based methods. Why not for food analysis and then tracking like food adulterations? You can see if we can see like varroa also. I don't know if this sample contained, but no, we can see bumblebee as well. Have been in the same flowers.
Bumblebee.
Bombus, yeah, it's bumblebee.
Oh.
So just because they have been in the same flower as the bee.
Yeah. Also, we can see like
that's crazy.
We can also see like DNA traces from insects that have produced the sticky liquid. Aphids, aphids. Do you know that the insects that are producing on the leaves of flowers sticky liquid, and honeybees are collecting it. Like they're plant pests.
Siis mitä?
Mitä se on kasvien lehtiin tulee sitä tahmia?
Ja kirvahunajaa siis ja like a forest honey. Yeah,
and we can see like traces from Galeria melonella. Do you know this is like kärjäkoi?
Mikä?
In Finnish you have the similar word like in Estonia, kärjäkoi.
Kärjäkoi.
Yeah, or vahakoi.
Vahakoisa.
Yeah.
Vahakoisa.
Yeah. Also traces from that because they have been in contact with honeycombs. So this kind of pests we can also see very often, like this kind of.
Thank you.
Yeah.
Very much. It's a wonderful day. Oli kyllä ihmeellinen reissu, aivan uskomatonta, että kuinka paljon sitä hunajanäytteistä todellakin nuppineulan pään kokoinen kasa jää jäljelle jotain töhnää. Ja siitä löytyy yli 10 miljoonaa DNA-pätkää ja sieltä pystytään erottamaan, mitä DNAta siellä on. Esimerkiksi sieltä löytyy kimalaisen DNAta hunajanäytteistä sitä varten, että ne on käynyt samassa kukkasessa, mehiläinen ja kimalainen. Aivan käsittämätöntä, että kyllä tulee väärentäjillä olemaan tulevaisuudessa vaikeat olot. Tällä labrallahan on kyllä kaiken maailman sertifikaateita ja oikeuksia tähän DNAn tutkimiseen, vaikkei hunajan aitouden tutkimiseen, koska tämä labra tutkii esimerkiksi kaikki Eestin ja ulkomaaltakin tulee niille näytteitä raskaana olevien naisten, että niillä on kaikki asiat niin kuin pitää siinä hommassa, niin taikka, että ja onko sikiöllä mahdollisesti Down-syndrooma tai jotain, niin tämmöisiä asioita, niin kaikki nämä verinäytteet tutkitaan samassa laboratoriossa. Nyt on tärkeää kuitenkin muistaa, että välttämättä ihan nopeasti ei tule tapahtumaan yhtään mitään radikaalia, koska tämä menetelmä on uusi. Täältä puuttuu tieteellinen vertaisarviointi, tieteelliset julkaisut ja auktorisointi Hunajan aitouden määrittämiseen. Ja ne on kaikki varmasti tulossa ensi vuoden aikana toivottavasti, mutta tällä hetkellä niin ei voida yhtäkkiä mitään hirveän radikaalia tehdä ja tulee aika iso vääntö tästä asiasta, että joudutaan vähän vartoilemaan vielä tätä uutta tulevaisuutta, mutta kyllä mä jo tässä kohtaa uskallan sanoa, että pistäkäähän jaokkeita tehdä lisää. Kysyntää on.